So welcome back. We had a good introduction to graph theory and the theory of networks in the previous class. Today we are going to focus on understanding the structure of these networks and particularly how they are applied to biological scenarios, and we will look at a host of network parameters which capture various features of nodes and edges and what they mean for the structure of the network and the importance of a given node or an edge and so on. Okay. So in biology there are many applications for graph theory. So there are many biological problems that naturally map back onto graphs. So some of them include pathfinding in metabolic networks or link prediction, right, to find if a couple of proteins are connected, or even in more complex knowledge graphs and so on. So predicting if there should be an edge between a pair of nodes, and then read assembly of various genomic data sequences. So when you have next generation sequencing data, you typically get that in the form of small reads. So how do you assemble them? And this involves some graph algorithms, and you will actually hear quite a bit about this in the other course in this module, where Professor Manikandan will be talking about some of these very interesting algorithms. And then how do you identify clusters or modules in large networks? I think this is something we will look at immediately in the next class and so on. Network biology today has gained widespread popularity. The review you see at the bottom of the screen is a very nice paper which I would encourage all of you to read. This is a very interesting paper that talks about various aspects of networks, various aspects of network structure, kinds of models that are used to capture networks. We will be discussing many of them in detail in this course as well. There are also many applications in ML and data science. For example, this starts with community detection, which is one very important aspect. A lot of applications of graph theory, if you will see, involve certain aspects of identifying what are the most important nodes in a graph, what are the important edges in a graph, or what are the important groups of nodes or subgraphs in a given graph. So what part of the graph is very important, has important functional roles in an organism or in the context of other networks, like it could even be an air transport network or anything, right. So which sections of the network are vulnerable or strong or important or essential for the function of the network, and so performance of the network and so on. Okay. So now let's look at some examples of standard examples of graphs. For example, Facebook, Twitter, these are all graphs that you are social networks that you are very familiar with, and all the way up to protein interaction networks and so on. So what are the nodes and edges in each of these? I would like you to think about these for a moment before I show you the slide and tell you what are the nodes and edges. So each of these you want to pick what is your node and what is your edge, and this gives you a particular ability to ask questions of the graph. Depending upon what you choose as a node and what you choose as an edge, in some of these things it's straightforward. I don't think there are too many choices. In Facebook, well, obviously people are going to be nodes and friendships are going to be edges and so on. You don't have much of a choice. But on the other hand, in say a protein structure, you can actually represent the networks in many different ways. Can you represent an atom as an edge, an amino acid as an edge, or some structural aspects as an edge, or whatever, right. So there are different ways of representation, and each of these representations enable you to ask different questions of the protein structure itself and so on. So we look at some of these examples a little later on also in the course, but for now let's just try to fix ideas and focus on what are going to be the nodes and edges in each of these graphs. So if you look at Facebook, the nodes are going to be people, the edges are going to be friendships. In Twitter, the nodes are typically people, well, it could also be businesses and so on. Well, that also applies to Facebook. But the edges are follows, and there is a definitive notion of direction to these. And we already talked about directed and undirected networks in the previous class, and Twitter is clearly a directed graph, whereas Facebook is typically an undirected graph. In a protein interaction network, again, it's similar to Facebook because two proteins bind, two proteins interact and so on. Whereas in a gene regulatory network, you have more challenges because you could have A activate B, B repress C, C activate A and so on. So you have these kinds of different behaviors that are not necessarily reciprocal, right. So A may activate B, B may have nothing to do with A. So there is, again, this is going to be an undirected, a directed graph. This is going to be a directed graph. Then you have metabolic networks where you have very interesting representations. We'll probably look at it at some point of time. And I think we also had a decent discussion of citation networks and co-authorship networks, and how some of these are, especially co-authorship networks or any co-starring network or so on, are better represented as hypergraphs or bipartite graphs. In a food web, you are going to have species as nodes, and who eats whom form the edges. And in a protein structure, you have different kinds of nodes and edges that are possible. So, can you actually use an atom as a node, or can you actually use an amino acid as a node, or C alpha atoms as nodes, and whenever two C alphas are too close to each other, you can think of them as an edge, and so on. So, all of these possibilities are very much doable. I mean, they are typically employed in these kinds of graph models, and each of them has different sorts of implications for what questions you ask and what do those predictions mean, and so on. And then you also have molecular graphs. We've used them in the past, and I'll talk about an application later on in the course, where it's relatively simple. Every node is an atom, right. So, let's say you have methane, then carbon and the 4 hydrogens are going to make up 5 different atoms, and there is a single edge between the carbon and each of the hydrogens. Right. So, there are 4 edges and 5 nodes in this graph that corresponds to methane. You can also take care of bonds that are into the plane and above the plane and so on, basically trying to account for optical activity, chirality and things like that. So, just to quickly recap, in Facebook the nodes are people and the edges are friendships. In Twitter the nodes are people or businesses and the edges are follows, which gives it direction directly. And in protein interaction networks, it's proteins and interactions, bindings or whatever. In gene interaction networks, the nodes are genes and there are regulatory effects. In a metabolic network, you have multiple types of metabolic networks, but typically you can have metabolites as nodes and the edges corresponding to reactions. This is what we would call a substrate graph. You also have citation networks where you have research articles as nodes and you have citations as edges, and there is a notion of direction here, right. There is always this network that goes in temporally one direction. You can only cite past papers, you cannot cite future papers ever. Right. And then you also have the food web where, or co-authorship, you also have the co-authorship networks where the nodes are authors and the edges capture co-authors, right. So, those who have co-authorships in a sense, or it could even be papers, but not necessarily. We did discuss some of these issues in the previous class. And then we have the notion of protein structure, where a simple one would be amino acids be the nodes and contact maps capture the edges, basically some sort of energy or distance between the different residues. And the next one is that of a molecular graph, where you have atoms for the nodes and bonds for the edges, and you could even have like edge weights to capture multiple bonds like C triple bond N or a C double bond O and so on. There are many kinds of networks in biology, and in fact the function of every living cell is a resultant of all these underlying complex networks. What are these networks? Typically we try to deconvolve them, but in reality they are all one complex mega network where all of these interactions take place. So, you have metabolic networks, you have protein interaction networks, you have gene regulatory networks, signaling networks which includes typically proteins, receptors and also small molecules called second messengers and so on. Of course, you have other kinds of networks like food webs or protein structure networks. These are network models that are used to study different systems. They are not unlike the others that I have highlighted. These are basically within a single cell, within every single cell in every single organism. And cellular function itself is a result of all these underlying complex networks. You typically see complex or beautiful pictures like what you see on the right of your screen, and these models or these networks help us ask and answer very interesting questions of biological systems.